Alright, here's Xingyu Lu going over the stuff which the professor talks about in the November 24th lecture. The guest lecturer is Dr. Cheng Zhu from the BME Department of Georgia Tech. His main research fields are the molecular biomechanics and mechanobiology. Biomechanics is the study of the structure and function of biological systems such as humans, animals, plants, organs, and cells by means of the methods and mechanics. And molecular biomechanics focus on the studies of the mechanisms of proteins, nucleic acids, and understanding of the molecular mechanisms of stress transmission, mechanosensing, and mechanotransduction in living cells. Dr. Zhu proposed that the cell is a collection of protein machines, and this machine runs at a very small scale. It then gave an example of mechanobiology at the cellular scale. The example of atherosclerotic plaque is given. These plaques tend to form at branch points and curves in arteries. They are hard to be diagnosed and detected in the early stages, but can be potentially detrimental in the late stage. They have a higher chance of forming fibers in shear flow than in steady state. Another example of mechanobiology is that endothelial cells can sense their environment and respond accordingly. Dr. Zhu also mentioned to us that not all cells differentiate to perform in a certain environment, so mechanical impulses might be very important in determining the path they follow when differentiate. Cells are able to sense mechanical signals in many different ways. The receptors could be cell organs, transmembrane proteins, or even other organs that are buried under the endothelial cells that are not directly exposed to flow and cell signaling. Next, Dr. Zhu introduced some biomoleculars that are related to cellular mechanical signaling. He gave an example on mechanopresenter, mechanoreceptor, mechanotransmitter, and mechanosensor. Mechanopresenter is a molecule that presents information to the mechanoreceptor upon in its mechanical environment. It is a specific protein-protein interaction which resemble the common cell signaling that we are familiar with. The definition of mechanosensor is given as a converter from mechanical signal to chemical signals. He then mentioned a special protein called von Willebrand factor, aka VWF. VWF is a blood glycoprotein involved in hemostasis. It is a large multimeric glycoprotein present in blood plasma and produced constitutively as ultra-large VWS in endothelium. When a person gets hurt and he starts bleeding, plasmates are diffused and blood vessels in human bodies are disturbed. Here, the VWF is released to modify the flow. Always VWS presents human body as an aggregate, and it needs to be cleaved so that it will perform its fully function. Some experiments were done to determine the binding domain of VW factor to other vessel cells. There are two pieces that account for the binding. One of them is the longer site known as TSP, and the other one is the CUB domain, which is short, relatively short compared to TSP. But the result turns out that the actual binding site is the shorter CUB domain. Finally, at Instagram known as ADAMST come into place to cleave the large VWS into monomers so that they can perform their functions. In this case, VWF is a mechanopresenter because it changes its conformation to present different sites. Now talks about the TTP microstrombin and other definitions that are crucial to GP1B induced intracellular calcium release, such as ideal, slip, and catch bound. Overall, he was trying to convey an idea that molecules have distinguished binding sites and affinity at different conformations. Some can be cleaved to be transformed into monomers or smaller scale proteins, and some can bind to each other or even other proteins to become a compound so that they can create a new binding site for the incoming signaling. Yeah, this is basically the idea for the lecture. So I hope you enjoy my introduction. Oh, by the way, this is the extra video for Physics 2212. So it's taught by Dr. Greco. Yeah, that's it.